Hello, my name is Marybeth Gilliam, and this is the Outperform Cancer podcast, where we identify anti-cancer strategies found in peer-reviewed scientific research. You may have heard about precision medicine. In fact, you may have heard about it on my show. But have you heard about functional precision medicine? It's an emerging science with the potential to be an incredibly powerful tool for oncologists and patients. Currently, when a patient's cancer progresses, the next treatment decision can feel surprisingly imprecise. An oncologist may have 3 or 4 medically reasonable options, each supported by evidence from a large population study. But the patient sitting in front of that oncologist is not a population. They're an N of 1. And so far, there has been no reliable way to know which of those treatments that individual's cancer will respond to or resist. And every unsuccessful treatment comes at a real cost. Time lost, unnecessary toxicity, and sometimes the loss of an opportunity to try something that can be more effective. This is the problem at the center of today's conversation. My guest is Ed Chow, Chief Science Officer and Scientific Co-founder of KYAN Technologies, a functional precision medicine company that is developing a very different way to help guide cancer treatment. Instead of relying solely on the genetic blueprint of a tumor, KYAN's Optum AI platform obtains living cancer cells from an individual patient, exposes those cells to different drugs, and uses a mathematics-based platform to predict and rank which single drugs and combinations of drugs may be most effective for that particular cancer. And the early clinical evidence is compelling. In a prospective study involving patients with relapsed or refractory non-Hodgkin's lymphoma, Optum AI platform-guided treatment was associated with get this, 44% lower risk of cancer progression compared with treatment selected by the physician alone. The median progression-free survival was 129 days compared with 79 days in the clinician-guided group. Now, the technology is not perfect, and to be clear, it does not replace the judgment of an oncologist. However, the data is compelling, and it raises an extraordinarily important question. Before we give a patient a cancer treatment, could we test that treatment against the patient's living cancer cells and reduce at least some of the trial and error? And in doing so, could we dramatically improve cancer outcomes simply by better utilizing the medicines we already have? It's an important question because it means that we're not waiting for a cure. This is about using what we've got at our fingertips today. And in this episode, Ed explains how that technology works, what the clinical studies have shown, where its limitations remain, and how patients in the United States can access the test today. Join me in welcoming Ed Chow. Ed, thank you so much for coming on the Outperform Cancer podcast this morning. We are really excited to talk to you.
Thanks for having me. I'm really excited to have this conversation.
I'd love to start off just addressing the fact that there's a lot of talk in the industry about precision medicine and personalized cancer care today. But for many patients, treatment still involves an incredibly frustrating amount of trial and error. And my understanding is that your company is trying to help address this issue. So I'm hoping you can start us off by describing the problem that your team is trying to solve.
Yeah, so KYAN Technologies, we're a functional precision medicine company, and we are essentially trying to figure out how to provide oncologists and patients with greater insight as to how all the different treatment options that are available to them may be sensitive or resistant in their cancer, in their specific cancer. This is getting increasingly more complicated as we get more combination therapies also coming into the clinic. And so where our platform really shines is the ability to evaluate all the different potential combinations that a patient might be given within a defined set of drugs that are available for their particular cancer type.
So I'm going to give you a scenario because this just happened to someone that I was talking to recently, a friend of mine. She had progressed, her breast cancer had progressed, and her oncologist came back to her, and there were four different options. And the oncologist said, "Hey, we can put you into this clinical trial. We can put you on in HER2. We can put you on." I think it was Lynparza was another option, and then there was a fourth. So if a patient is presented with that option, like this friend of mine was, you know, she called me and said, "Well, how do I choose?" Can you talk a little bit about that? Like patients facing this, is your company designed to help them in the face of those kinds of decisions?
Yeah, exactly. So oftentimes, this is how it's essentially started was when we started, there was this idea that for many of these cancers, you are patients and doctors are typically presented with these three or four choices. And one of our clinical partners described it as dealer's choice, right? It's like, you might get whatever hand you get. You kind of know that the different overall response rates across them are about the same. So whether you go with one or the other, at a population level, they know that, certain percentage of patients will respond. It doesn't matter which one you give. But for the individual patient, it does matter, right? And so the
Right.
idea is for the individual patient, can we provide greater insight, even though clinically it's dealer's choice in over 100 patients, you'll probably get the same number of patients responding to each one. But can we, for that particular patient, gain insight, say, no, they are better off with option A instead of option B, C, and D? Or are there other options that are not even being considered? And should they just even skip all four options and go to, what would normally be third or fourth or fifth line treatment options instead?
This to me is the heart and soul of precision medicine. And I've just recently learned that it's being called functional precision medicine, which is interesting to me because, I've been talking a lot about precision medicine and that has gone hand in hand with biomarker testing. So we were talking about, okay, we're going to understand the drivers of any mutations. We're going to get biomarker testing from companies like Natera or Boston Gene and really understand the cancer. But what I hear you talking about, I'm hearing this new title for, which is called functional precision medicine. And it seems like it's the second half of this. It's like, okay, we can understand the cancer, but now we need to understand how the cancer is working in real time with medicines. Am I accurately describing that?
Yeah. So essentially, what we see right now with precision medicine is it's largely driven by biomarker testing, right? And as you said, it's oftentimes specific driving mutations in cancer drive cancer pathogenesis. And thus, we've developed a lot of drugs around those. So, as you mentioned, like EGFR mutation is a great example in lung cancer. And so, we've developed drugs to target EGFR mutations. So, if you get the biomarker testing and you come up as EGFR mutant positive, then typically you'll get that EGFR drug. Same with breast cancer with HER2. And now, we're starting to see KRAS inhibitors with specific mutations. But at the heart of it, these are still individual mutations, and these are still drugs that only target a specific mutation. But cancer is complex. Biology, human life is complex. There's oftentimes not a single gene for most cancer types. There's not a single gene that's driving all of cancer. And every day we're learning new molecular mechanism for how cancer or even biology in general works that goes beyond just the changes in the genes, right?
Yeah.
And so the idea at the heart is typical biomarker testing is just a snapshot. But functional testing is what is real life, what is really happening. It's kind of like the movie versus the still picture, right? And
Yeah.
at the heart of it, it's really, if you want to know how your cancer cells are going to respond to a drug, give the drug to the cancer cells and see how they respond. And how they respond will likely have a greater insight into how they'll respond inside the patient. And then just dealing with the complexity is then where we pick up.
Okay, so that is a terrific lead into a deeper dive into. So, what exactly is KYAN Technologies doing? Help explain for the patients and the physicians out there that are new to your company and new to this concept of functional medicine. What do you do?
Yeah. So essentially, what we do is when a patient comes in, wants to get access to our test or a test like ours, essentially the first step is to work with the oncologists and the surgeons to collect a live tumor specimen. So that is something that's unique to functional testing, is that we require live tissue or a live sample in case of hematological malignancies or blood cancers. So we require that live sample so that we can get the live cancer cells. And then once we get the live cancer cells, we put it onto our drug testing plates. And for KYAN Technologies, our unique platform is that we use math-based testing. So we use math principles to design how we test the different drug options in combination with each other, with the idea that by using math-based approaches, you don't actually have to necessarily test every single possible combination to know how all possible combinations are going to do with a certain patient's cancer sample. And so then we get a quantifiable phenotypic output. So we measure drug response in these plates in some type of quantifiable manner. And then that goes into our analytics platform. What our analytics platform does is then assigns each drug, as well as every drug-drug interaction, some type of mathematical value, and then builds a ranked list of all the different clinically possible treatment options from the best to the worst. And then from there, we can pull out, these are the options that might be effective for the patient. These are standard of care options that might be effective or are definitely resistant for that patient. And then we provide that as a patient report for the oncologists and the patients to make a more informed decision. So that then they'll say like, oh, standard of care option A, maybe is borderline effective, but standard of care options B, C, and D are definitely not. But, oh, there's this other option that is currently in clinical trials or has been clinically trialed that is even better than standard of care option A. Maybe I should talk to my oncologist about going with that treatment option.
To me, this technology is really astounding, and it solves such a huge problem for the patient and the clinician. I mean, both want to see the patient thrive and do really well. And it sounds like because you're using fresh tissue, you're able to understand how it's working because you have the full tumor microenvironment. Is that true?
Yeah. So I will say that these ex vivo culture systems are not perfect. So they don't fully recapitulate the tumor microenvironment. So I will say for solid cancers, we do mix all the cells that were present in the tumor. For blood cancers, it does get a little more complicated because for the blood cancers, oftentimes, for instance, with like leukemia, the leukemic cells represent only a small percentage of the total blood or the total bone marrow aspirate. So we do isolate the leukemic cells first from the sample and then treat directly with the leukemic cells. So it isn't fully recapitulating, say, like the bone marrow microenvironment that AML might sit in the bone marrow or in the peripheral blood with all the other immune cells that are present in the blood circulatory system. So doesn't perfectly recapitulate, but the idea is that it's good, but maybe not perfect, but it's still better than what is currently available.
Right. Well, and I think that's a big thing, right? I really appreciate the engineering approach. So when you're talking about applying, math to this to use as a tool, it makes a lot of sense to me, and it's actually very encouraging to me as a patient that we're starting to take these engineering and mathematical principles and apply it in medicine. I love to see that, and I can understand that it's not perfect, but something that I know from when I was working in engineering is that iteration is part of the key, right? You iterate, you make it better, you understand what's happening, and then you see the next step. And I would imagine that's probably what you're doing with your own testing. Is that true?
Yeah, so exactly. The idea is that we start from a core engineering based principle. So at the heart of our platform is the idea of how do you use engineering principles to more efficiently determine how different components should come together to create your desired product. And so we apply that to the biomedical space. But as you mentioned, technology is constantly improving. These ex vivo cell cultures are constantly improving. How we measure drugs in cell culture are constantly improving. And we can always keep onboarding these new modalities of ex vivo cell culture and ex vivo drug testing to constantly improve our system. And we're doing that now. So for instance, right now, most of our CLIA LDT reports involve small molecule drugs, so chemotherapy and small molecule targeted agents. But now we're incorporating more advanced cell culture systems and analytics to now allow for immunotherapy screening. And we're doing those clinical studies now. And that's kind of that iteration to improve and expand the types of drugs and the accuracy in which we're evaluating those drugs keeps improving.
That's pretty awesome. We had spoken earlier, and it had been explained to me that for each patient's tumor, you can set up a battery of 12 different drugs tested at 3 different dose levels, and that's what you run the sample against. But what was also interesting to me is that it's not just the 12, and you have, I don't know, like almost 200, I think, different drugs that are standard that you could pick from, for any patient could pick from, that might be relevant for that patient. But also that you could do things that maybe aren't on your list today, but that you could still test against. So, for example, if someone wanted to see if an off label substance that might not be a chemotherapy or even an immunotherapy, they wanted to see if an off label substance would work, you would be able to test it. Is that accurate?
Yeah. So definitely. So as you mentioned, right now we have over almost 200 FDA approved clinical drugs in our current inventory that doctors and patients can choose from to test. Typically, we run the test in batches of 12 drug panels. Within the 12 drug panel, we get all the possible different combinations that are available. So like less than 200 experiments in a single drug screening plate. Ultimately, if you look at 5 drug combinations and small less, it's just over 1,500 different combinatorial options that are potentially clinically actionable for any given patient sample. And then, as you mentioned, if there are drugs that are not present in our inventory that the patient or the doctors really want to test in these panels, as long as we have enough lead time, and typically we say if we can get a 2-week lead time between when we're notified and the surgery, we can usually get the drug in time to do the test. Otherwise, we may have to cryopreserve the sample until we get the new drug. But definitely for certain rare cancer cases, we've onboarded new drugs. That the clinicians and the patients were interested in evaluating for some of these rare cancers. We've done it multiple times now to good success.
I get so excited about this because this is new data that patients have not had access to before. And I really believe in, follow the numbers, right? What is this telling you? So speaking of numbers, can you tell us a little bit about your results? So you run this, but as you were saying earlier, it's not a 100% situation at the moment. There is still some variables that you can't account for. So what is the accuracy of the test currently, and what could a patient expect when they get this test done?
Yeah. So, we've developed this in academic research, medical research institutions. So we've run several prospective clinical studies. We've also done retrospective studies as well, using our real world data. So as a clinical laboratory, we've been running the tests in Asia for just over 3 years. And across 600 samples tested, more than 600 samples tested across 20 different cancer types. If you look at both our retrospective and our prospective clinical concordance studies, we're averaging about 86% total clinical accuracy, depending on the cancer type. So some range in like the low 70s to like the upper 90s. But if you look at all 20 cancer types in aggregate, it's on average about 86% total clinical accuracy.
Well, still better than, hey, we got 4 options. Let's just
Yeah,
pick.
yeah,
Yeah,
definitely.
so it's still very impressive. Is it easier, or do you find results are more accurate with a leukemia or a blood cancer versus a solid tumor, or how does that influence the results?
So I will say that where we find the accuracy is oftentimes in primarily how well the drugs are for that different cancer type. So I will say that the better the drugs are, also more targeted agents tend to have, if the cancer type tends to use more targeted drugs, we tend to get higher clinical accuracy just because of how dirty cytotoxic chemotherapy drugs are. So I would say that that's the primary differentiator. Um,
And when you say dirty, do you mean that they are diffuse and they just are too broad spectrum?
Yeah. So I would say that, like, for instance, with chemotherapy drugs that generally target general molecular biology mechanisms. So, for instance, like chemotherapy typically is this idea of stopping cell proliferation, right? And it's not necessarily specific to individual cancer types, but just over the overall idea that cancer is a disease of uncontrolled cellular growth. So those tend to be on the lower end in like the slower 70%. Whereas targeted agents, which target a very specific driving mutation or
Mhm.
combinations or a very specific cancer mechanism that is highly unique to that cancer type, you tend to see those, the accuracy, goes up to into the 90%.
So that kind of makes sense to me, just as a patient, that if you have a targeted drug, it's either going to work or not
Yeah.
work for a person, and you're able to just kind of sort that out with a little more accuracy than something that's like
Yeah.
more broad spectrum. That makes sense to me.
Exactly. Yeah. So for
Mhm.
instance, if you have a HER2 positive breast cancer, it's either going to work or not work. And if it's working, you'll see that activity. And if the patient has progressed to a period where now they're no longer sensitive to that specific HER2 treatment,
Mhm.
it's more likely to show up in ex vivo drug testing.
So let's dig a little more into your research, because I try to help patients understand, and I try to talk to people who have uh, done peer reviewed research. So they've been evaluated, they're doing clinical trials, so that patients can, you know, understand where a new technology is on the continuum. Just brand new, not a whole lot of evidence, or gee, this is got a substantial amount of evidence, and we're moving towards bringing this forward to more and more people, like a phase 3 and ultimately standard of care. Can you talk about where your evidence is on the continuum, and maybe one of the tests or trials that you've conducted, and dig into that a little bit, the rigor of that trial?
Yeah. So one thing that KYAN Technology is really proud about, and obviously, I feel really fortunate having originally developed this technology out of an academic medical research institute, is that we have quite a lot of prospective clinical studies completed for our platform, um, including everywhere from non-Hodgkin's lymphoma, AML, soft tissue sarcoma. And we're still actively engaged in a number of clinical studies right now. So currently, right now, we have clinical trials for glioblastoma, ovarian and endometrial cancer, breast cancer, pancreatic cancer, colorectal carcinoma. Um, So we're constantly generating new clinical evidence for our platform. I would say the strongest peer-reviewed evidence that we have is probably in the refractory relapse non-Hodgkin's lymphoma setting. So that was a prospective study that we published multiple papers on, but the final paper was published in 2025 in JCO Precision Oncology. So that was our prospective study in 105 refractory relapse non-Hodgkin's lymphoma patients, split about half-half between B-cell lymphoma and T-cell lymphoma. In that, we saw an overall clinical accuracy of 75% across a wide range of treatment options that were given to these patients. So it wasn't tagged to a specific single treatment, but it was whatever they were given in their treatment journey, from anywhere from 2 lines of prior treatment all the way up to the 7th line of treatment. But more importantly, that study also showed the clinical utility of functional testing because we saw significant improvements in progression-free survival with a hazard ratio of 0.56 if you compare platform-guided versus clinician-guided. And then another metric that people are using in prospective studies for precision medicine is this idea of PFS ratio, or progression-free survival ratio, where they're comparing what is the progression-free survival time of the guided treatment versus the patient's prior line of treatment. So the assumption is if you're accurately choosing the right treatment for the patient, then the amount of time that they benefit or you can stop cancer from progressing should be longer than the prior treatment where it was not guided.
I want to just go back to something you said earlier, because you used a statistic that I'm not sure that everyone will understand. You were talking about the difference between kind of dealer's choice, physician's choice versus your technology. And I think you said it was kind of about 0.5 or 0.05 or so. Can you tell me what that
Oh, yeah.
statistic?
Right. So hazard ratio, this idea of hazard ratio is if you compare, say, one treatment decision-making platform versus, say, another treatment decision platform, the 0.56 suggests that there's about essentially like a 40% improvement in clinical outcome if you go with one route over the other. So the idea is that if the hazard ratio is 1, then that's really dealer's choice. It doesn't really matter what you go with. Like, one will be the exact same as the other. And then say if your platform is below 1, the lower the hazard ratio value is, the more clinical benefit you get going down this platform versus the other. Even with drugs, right? They'll say, like, comparing standard of care to like a new drug that's getting developed, if you can show a hazard ratio of 0.56 or 56, that's fantastic. Then you're showing that for, 46% of the population, they'll definitely benefit using this new investigational drug over standard of care. And so typically we use that to measure clinical benefit. If you can really move the needle, that far, then, it's a good thing.
So just to sum up then, you're saying that you saw a slightly more than 40% improvement over just the physician's choice.
Yes. Yeah. Yeah.
Because that's big.
Yes. Yeah. Yeah.
It's, it's
Yeah.
important. That's important.
Yes. Yeah.
And I know that you had mentioned that you're working with several different cancer types. Some of them are very challenging to treat. Glioblastoma is one of those. I lost 2 friends to glioblastoma this year. It is a terrible disease. Neither of them, unfortunately, neither of them made it a full year after being diagnosed. You mentioned that you have done some testing with glioblastoma patients. Can you tell me about that test? Because I believe you looked at likely responders for first-line TMZ, which is one of the standard of care drugs, plus radiation. And if you could elaborate on that and tell us, what did your test find?
Yeah, so we're currently conducting our glioblastoma clinical study right now in Singapore out of in collaboration with the National University of Singapore Hospital. And so we just completed our interim analysis, which we'll be publishing very soon. But essentially some of the interim analysis that we saw was if you compare our platform predicted responders and non-responders to first-line STU protocol, which is the temozolomide with the radiation therapy, we saw improvements of almost 5 months difference. And the cool thing is that we were able to then also compare how our prediction does compared to MGMT methylation status, which is the standard of care right now. So many patients right now will get the MGMT methylation assay done, and that predicts sensitivity to temozolomide, where if you're MGMT methylated, you should have greater sensitivity to temozolomide. And in that, we saw that definitely the MGMT methylation status does correlate with temozolomide sensitivity. So patients who had MGMT methylation, we saw were at about 17 months of progression-free survival versus unmethylated was 11. But in our platform, we saw 22 months versus 7. So there was an even greater difference in predicted progression-free survival. We were both able to identify both the true non-responders who are really not going to do well, as well as also potential long durable responders to the first-line STUP protocol.
Wow. Yeah, impressive, really impressive, especially with something like glioblastoma, where every month counts, and being able to double survival or triple survival is a really big deal. So let's talk a little bit about the technology and how it came to be developed.
So the core technology, the math-based approach, was initially developed at UCLA. It was started with one of our other scientific co-founders, Professor Chi-Ming Ho, who is one of the top engineering professors in the United States for many, many years. He's considered one of the fathers of MEMS technology and microfluidics. And it was really him that he was the one who really pioneered this idea of taking engineering-based principles and applying that to biomedical research. So he had an expertise in complex system analytics and black box engineering. And he used these math-based approaches, various different math-based approaches to solve how can we model biological systems without actually knowing what all the different switches and circuitry happens in a cell or in an animal or in a human. And the idea is that by using engineering complex system analytics, you don't necessarily need to know every single gene, every single mRNA, every single protein that's working to do its thing, that you can basically model it out using math. And then using that model, you can then basically predict how different drugs or inputs will affect different outputs like cell death. So at UCLA, it was primarily developed in the context of infectious disease. At the time when we started working on this, I was a graduate student at UCLA working in a microbiology lab that was doing antiviral research. And then for me personally, due to personal reasons, I was touched by, my dad got cancer when I was in graduate school. And so that part of that life experience caused me to switch to cancer research for my postdoc and to start my own academic lab in Singapore doing cancer research. And so as I started moving into cancer, then, I continued to work with Chi-Ming Ho and one of our other scientific co-founders, Dean Ho, on this platform, but I applied it very specifically in the cancer setting. And then as we started to develop it, that combined with increased expertise and the use of patient-derived models of cancer, we started getting closer and closer to the patient to the point where we were running the test on patient samples, and we saw that we could actually compare how different treatment options might work given a patient sample. And then, once we tried our first patient, who happened to be a hepatosplenic T-cell lymphoma patient who had failed 7 lines of treatment, so at the time, we were using patient samples to identify novel combinations that might be more effective for cancer. But in this case, they said this patient has no more options. So we're going to go to hospice for this patient unless you can identify another treatment option. And so we ran the test on this patient. We actually identified a combination that the Singapore hospital system had actually clinically trialed 2 years before we ran this test. And so the clinical trial wasn't amazing. It saw about a 20% overall response rate, but this particular patient happened to fall within that 20% in our test. And they had a fantastic response. Even though they had already failed 7 lines of treatment, They saw this combination worked immediately for this patient within 2 cycles. And this patient went on to have a durable response of 18 months.
Wow.
And so based on that success, then we just kind of kept going with it and testing more patient samples, more cancer types, until where we are now.
Oh, I love those kinds of stories because it really hits me right in the core of my gut, which I just believed so much when I was diagnosed metastatic and I was trying to figure everything out. It's just the importance of getting the right medicine to the right person. Finally being able to look at an actual how will this work within this person and finding the right combination or the right drug for that particular person. It's everything. It's just everything. So I thank you so much for the work you're doing. I'm going to speak on behalf of patients. I really do thank you for all that work. I want to bridge now all of that work and that theoretical turn to practical, turn to functional that you have now and connect the dot to the patient. Because there are going to be patients out there and physicians out there that are like, well, this is really interesting. How do I get it? What do I do? Can you talk to us a little bit about, how do we connect those dots?
Yeah. So if a doctor, if an oncologist or a patient is interested in accessing our test, essentially what happens is, a week or two before the surgery. So it has to be, we have to get notified before the surgery happens so that we have an opportunity to collect the live tissue. But if they can notify us, a week, well, depending on where they are, for instance, if they're in, for instance, North Carolina, Vancouver, or Southern California, we can probably do like a 48-hour turnaround if they notify us, 1 to 2 days before the surgery. But ideally, 1 to 2 weeks before surgery, they can either contact us directly. Additionally, we also work with various concierge precision medicine groups to help with collection of the sample. And so they can also work with these concierge groups as well. And then once they notify us, then we start the logistics machine. So we coordinate with the surgeons and the pathologists if necessary on sample collection. The patient has to sign an informed consent form that they agree to use to the test. And then the oncologist fills out the requisition form with the relevant clinical information related to that case. And then they can also, the doctors, have an opportunity also to fill out our drug panel selection form, where if they have an idea of certain drugs that they want to test, they can inform us. Otherwise, we can also help with drug panel selection as well, depending on the patient's history, or we have also standard drug panels to just run off the shelf for various different cancers. And then once that happens on the day of the surgery, our logistics team or the partner concierge logistics teams will collect the sample, put it into these specially designed cold chain transport shipping boxes that keep the sample at below 8 degrees Celsius for up to 96 hours. And then they're just shipped to our CAP-CLIA lab through cold chain shipping, which is, fantastic now that because of COVID and mRNA vaccines, cold chain transport has been, completely redeveloped and improved. So you can get anything anywhere in the world in refrigerated very quickly now.
Wow. Okay. So I'm going to dig into a couple of those things, just clarify a few points for people. You mentioned there are 3 areas in the United States that you could do turnaround faster. Why those 3 areas? It wasn't New York City. So
Yeah. So,
so
so the real answer is because, we have our KYAN Technologies US staff themselves are physically based in North Carolina, Los Angeles, and Vancouver. So in those 3 sites, our staff can very quickly turn around and work with the local hospitals on sample collection and shipment. Outside of that, we can either work directly with the hospitals outside of those areas, or we can also work with concierge precision medicine groups that specialize in logistics of other hospitals in other states for sample collection.
okay. So you have some US presence that can help facilitate.
Yes.
If someone wants to contact you, is there a through your website, or can we do that through your website?
Yes, exactly. So if they want to directly contact us, there's a contact us form on KYANtechnologies.com that they can contact, and that's probably the easiest way that they can get in touch with us directly. Yes.
Great. We'll put that into the show notes. And for everyone that's listening, KYAN is spelled KYAN, KYAN Technologies. So let's say someone has contacted you, they've set up their fresh biopsy that's being shipped to you. That's great. What happens on the other end? You finish this and someone gets a report. Can you describe for us what's in that report?
Yeah. So for our patient report, as I mentioned, even though what we get is, in terms of potential clinical combinations of there's like over 1,500 options, we're not going to info dump all 1,500 options. So what we do is we provide tables that are easier for both the oncologist and the patient to read. So the first table are all the available standard of care in that drug panel, both single drug and combinations. If it's standard of care for that particular cancer, then it goes into table 1 in order of best to worst. So for instance, if it's breast cancer, so any single drug or combination for breast cancer that's in that panel is presented in table 1. Table 2 then are any drug combinations or single drugs in that cancer type for which there's a peer-reviewed published clinical trial or clinical study for that combination. So for instance, if it's a combination that has been clinically evaluated in breast cancer, it'll go into table 2. And that patient report will also have a hyperlink to that paper so that both the patient and the oncologist can quickly access that paper that we're referring to, and they can look at what are the clinical outcomes that the clinical trialists saw, what were the adverse events or toxicities, and what was the dosing regimen that they used for those. Then table 3 are any drugs or drug combinations for which there's a peer-reviewed clinical study in some type of cancer. So if we're using breast cancer as the example, maybe this combination came up as being evaluated in colorectal cancer. That combination would be in table 3, but it would have a hyperlink to the colorectal cancer study. So then the doctors can infer, even though it's a breast cancer patient, maybe I can use the dosing schedule that was used in the colorectal cancer study, and what were the toxicities and adverse events that were observed with this combination. And then table 4, the last table, are any top-ranked potentially effective combinations for which there's no peer-reviewed clinical publication at all. So these are potentially combinations that either there's no published data yet, or these are combinations that have never been tried in humans before. And so these would be truly novel off-label treatment options that are given to the patient. And then on that first page, we also provide a visual graph of all those different options that we put in those 4 tables. Additionally, we also have an appendix which shows, the single drugs in the panel in order from best to worst, as well as also the top 20 2-drug, 3-drug, and 4-drug combinations that came up in our analytics. But the meat of the decision is primarily in that first page with those tables.
Yeah, that's what I thought that you guys did a very good job of making it readable, understandable, and by separating the different tables, it was very clear the uptick in risk that someone could see as you move through the different options. So I thought that was really well done. Now, this is not currently covered by insurance. Is that correct?
That's correct. Yes.
So, are you working on that?
Yeah. So, we are currently working on that. I will say that for certain private medical insurance, I will say outside of the United States, some of the private insurers are much more amenable to reimbursement for medically necessary tests. One thing we are trying in the US is that there are certain states that have state laws for medical reimbursement of medically necessary biomarker testing, where they do define ex vivo pharmacological response as a biomarker output. And
Ah.
so for those, technically the state law says that insurance companies have to reimburse for functional tests like ours. But how you define medically necessary is still up to the insurance company. So in those situations, for those states, like California is one of them, where we are highly interested in patients from states like California or Arizona, where we can potentially work with the patients to
Mhm.
submit a clinical and scientific justification for the test for reimbursement as a medically necessary test as defined by their specific state laws.
That's fascinating. I actually was helping the American Cancer Society get those laws passed in my particular state. I'm in Vermont. Unfortunately, it's not passed yet. We're still working on it. But the law has been passed, and I believe it's like 23 states, 23, 26 states, something like that. So about half the country. But it is something that is constantly evolving. And so I do encourage patients to keep a lookout for that and to see what you can do as a patient to help by writing letters and communicating with your leadership in your state to make sure that things like this are um, are accessible to all patients. Now, if it is not covered by insurance and someone has to pay out of pocket, can you tell me what those costs are?
Yes. So currently, right now, our main CAP CLIA lab is in Singapore. We're hoping to onboard a US CLIA lab by the end of this year. But for now, if they want to access the test today, the sample does have to go to Singapore. So as a result, what we do is we charge an upfront shipping fee of $1,500. And then when the sample gets to our CAP CLIA lab in Asia, we run a QC analysis. And so it has to pass certain quality control specifications in order to ensure that we're most likely able to generate the test. So if it passes two QC specific requirements, our success rate of generating a patient report is 98%, over 98%. And so at that point, then we build the remainder value of the test, which is 4,000. So it's 1,500 upfront. And then if it passes QC, then it's another 4,000. And then we'll typically return the patient report back to both the patients and the oncologist within 7 to 10 days.
Wow, that's pretty fast. That's amazing. And I know that's a lot of money for a lot of people. But in the realm of what cancer care costs, recognizing that, nowadays the 1 month of a cancer drug like Verzenio is costing about $17,000 a month. The $4,000 to make sure you're actually getting the right medicine that's actually going to work seems like money well spent. So
Yes.
hopefully
Yeah.
this is something that will be covered by insurance. Now let's go to the other barrier. We've got the barrier of the costs. So we got the barrier of insurance. We got the barrier of getting the sample. Okay, last barrier is finding a doctor who will work with the report. How is that going? Are you finding many doctors that will and how does a patient go about finding a doctor that might be interested in this? Do you have any guidance?
So I will say that we've been fortunate to have clinical champions for the test. And then additionally, there are also concierge precision medicine groups that can also help guide patients to doctors that are open to the functional test. So for instance, ourselves, one clinician in the Southern California area that they can also go to for support both before and after using our test is Dr. Santosh Kesari out of Astra Health in Los Angeles. So he's a very good oncologist, faculty at UCLA, who can provide that patient guidance both before and after using our tests. And then there's also other concierge precision medicine groups such as Palitra and Kernus that can also help both before and after testing to guide patients towards clinicians and medical groups that can put those reports into action.
That's great. The companies that you were just talking about, Kernus being one of them, those that is a one of Sid Sibrandy's companies. Is that right? I interviewed him recently, and I have great affection and respect for what he has done for the cancer community. His foundation is also has a group called the Future of Cancer Care Today. And they are doing an incredible job. Anybody can contact them for free and get some guidance on how to utilize the latest technology for their own benefit, for their case. So, so I encourage people to go to KYAN Technologies, but also check it out. Future of Cancer Care Today and Kernus as other organizations that could help with this test.
And they also just recently added Palitra as well, Palitra, to that group, to Sid's group as well. And Palitra is is primarily focused on just the functional testing. So from that pipeline, Palitra can also provide additional expertise, not just for our test, but for functional precision medicine testing in general. And then also how to best use that report afterwards.
Fantastic. Great resources. Thank you very much. We'll make sure that those links get put in the show notes to anyone who is interested. Can you tell us a little bit about a real patient case that you might have had that demonstrates what this test can add? Just to kind of highlight for and bring back to reality for people that these are real people with real cancer problems that are getting helped.
Yeah, I mean, so I would say my favorite case is there's one case which was part of our refractory relapse non-Hodgkin lymphoma study. So I had the pleasure of meeting the patient twice during her journey after using our test. So she came in after going through the normal standard of care for DLBCL treatment. She came in the third line setting. We predicted that she would not respond to standard lymphoma salvage regimens. And we actually identified a breast cancer combination of everolimus and palbociclib for her. And so at the time, there was scientific evidence for potential anti-cancer activity of everolimus and palbociclib separately in B-cell lymphomas, but they have never been clinically trialed. But because of the high response rates of salvage regimen, they still went with the salvage regimen, but the hospital tracked her closely given that they had the patient report. And when it was clear that she was not responding to the standard salvage regimen, they switched her over to the breast cancer combination based on the breast cancer clinical studies. And this was just before Christmas of 2019. And even in 2024, as I was leaving Singapore and moving to Los Angeles to start expanding KYAN's offerings into the United States, she was still in remission. And, that one, really was really great for me because I could see that journey from when she first came to us and she was very weak. She couldn't drive. She had difficulty getting out of bed. She couldn't do the daily activities she wanted to do. Like she was very into art. She loved, walking to her near local market to get food, to cook for her family. And then by the time I left, Singapore, she was driving. She was doing her art again. She was living a full normal life. And just seeing her recover and get back to normal, I think it's just is the greatest benefit of being in this, starting this company and offering this test is just, and like you said, I think for us, it's just, how can we give patients more time? I just want to give people as much time with, their loved ones as possible, when they do have cancer. That's just one of many cases that we've seen of these responders. And the thing I love now about KYAN is that this test is now happening, every day, every week. And there are more and more of these cases for which, I'm no longer as deeply involved as when I was a professor, because now we have, a commercial team, we have lab staff, and they're constantly processing these samples, and these clinical outcomes are happening outside of, the specific clinical collaborators that I started with. And, just hearing these stories is, makes all the work worth it, and keeps us driving to how can we offer this test to even more people.
I love it. And I keep thinking, I have this picture in my head of your dad looking down on you and just being so proud. And yeah, that you're doing really, really important work here. I had a thought as you were talking about, patient, and that was that, sometimes with metastatic patients, their cancer is different in different parts of their body. I know I've met someone who was triple negative in one organ and triple positive in another organ. Can you deal with that, or do you have to have two different samples? How does that work?
Yeah, so we have looked at that. So, for instance, in our soft tissue sarcoma clinical study, we did include in that paper an example of just this. So, in that, there was a soft tissue sarcoma patient who had a brain lesion and a liver met. And at the time, we could only get access to the liver met initially. And so for that patient, we were able to identify a effective treatment option for the liver met, but unfortunately, the brain lesion did not respond to the drugs that was identified. But through their cancer journey, ultimately, the brain lesion did progress to the point where we were able to get a sample. And then when we were able to combine the reports from the liver met and the brain lesion, we were able to find overlapping sensitivities that then gave that sarcoma patient, I believe, another 7 months of response time for both sites. Having multiple cancer sites in the patient does complicate things, but I would still say that functional testing is still a step forward from, static biomarker NGS testing.
There are other tools in our toolbox, maybe radiation or something can be
Yeah,
used to, stereotactic radiation can really,
yeah,
be an incredible tool. There's the new technology in the reflection machine now that actually can track a tumor because it's the technology of the radiation plus married to a PET scan. So it tracks the tumor even in the lung, which is moving when you're in the machine. I mean, doing some
yeah.
really phenomenal stuff.
Yeah, I mean, definitely, even with the complexity of cancer, there are treatment modalities that can help, like you mentioned, radiation therapy, antibody drug conjugates have, what's called bystander effect,
Mhm.
where, they will kill the cancers they're designed to kill, but because of the radiation or the localized distribution of chemotherapy drug, it'll also kill other cells that are biologically different, but are still within that localized environment, and they'll still have that bystander effect to effectively treat those cancer cells as well.
Does a barrier exist with moving this forward to standard of care? Um, is it just, hey, we just need to keep doing more research, we just need more cases? Are there any other barriers that you see that are preventing this from becoming standard of care for everyone?
Yeah, so I would say that the biggest thing is that ultimately we have to show that functional testing as a whole, as an industry, in general, we have to develop that clinical evidence as a community that it can outperform current standard of care. And then once that happens, with these increased clinical evidence, then we can submit that to, for instance, like NCCN guidelines or ASCO guidelines. And then once it goes into those guidelines, then that's where it can really start moving the needle as standard of care.
I have seen that there are other companies pursuing this line of work as well. There's this technology. It is an exciting new category in addition to what you're doing uniquely. So, could you just talk a little bit about how you might be different, or what you're doing versus what else you see in the world?
Yeah. So our platform is uniquely different in that we are really focused on evaluating and comparing and ranking combination therapies. So I think it's all really two differentiators. One is the ranking system, where we definitively rank all possible treatment options within the drug panel from the best to the worst. And then two is the ability to test all the different combinations within that panel without physically testing those combinations. Because once you start thinking about 3-drug, 4-drug combinations, not just the doublets, the 2-drug combinations, it gets really complex. And with a single, you know, resection or biopsy, it's impossible to test all those treatment options. And so oftentimes with other platforms, you're typically having to target, specific standard of care combinations or specific combinations that the doctor is interested in, or specific treatment options. Whereas for us, it's more about, within that drug panel, what are all the possible combinations that you can test? And we can provide that information in a ranked list from best to worst. And I think that's where we're most differentiated. But at the heart of it, it's all still, a step forward from this static image. They're all, these more like live movie images of how your cancer is actually doing.
Yeah. I mean, to me, I love the numbers, right? Because they give you a solid ground to stand on. I don't know how to evaluate, hey, here's four options. Pick one. But when I have a rank order, like when I look at your report, it is very clear.
Speaking of that, the numbers also has increased value, especially for clinicians and patients who are used to seeing our report. The numerical values also give you insight into how the individual drugs within the combination are contributing to that combination. And so
Oh,
oftentimes you can also see that even if a combination comes up as top ranked and potentially effective, you may also see that it's almost all driven by a single drug. And the oncologist can then also make that decision. Is it, from a safety point of view, is it more worthwhile to just give that single drug? Or if it's, say, like a known 3-drug combination, but the doctor's worried about the toxicity of having all 3 drugs, it'll also give you insight as to the influence of, say, 2 out of the 3 drugs and say, maybe I can just give 2 out of the 3 drugs of this combination and still get the same effect, but with lower toxicity, lower cost for the patient.
I love it. Wow. Yeah, that's amazing. I think that is absolutely amazing. I know I've taken up a lot of your time. I have one more question, Ed. I'd like to end thinking about the impact of your technology on both the oncologist and the patient. Both parties want better tools to help patients outperform cancer, no doubt. The oncologist is responsible for safety, as you were just talking about, and the patient is responsible for their own life choices. What do you say to an oncologist who believes this is too experimental to influence a treatment decision? And what do you say to the desperate patient who sees a highly ranked combination and assumes it must work? You've got two sides to that.
Yeah. So, at the heart of it, the test itself is not forcing the oncologist to make any specific decision, right? It's giving them additional information to help them make that decision. And so I still always recommend that patients talk to their oncologist, and that at the end of the day, their oncologist is the person that they should listen to in terms of what is really safe versus effective and how to balance that. But I will say functional tests like ours provide additional information for their doctors to let them know both what treatment options should they just completely discard. So I will say the negative predictive value for our test as well as others is quite high. Those are typically in the upper 90%. And it's definitely removes potentially treatment options that basically take time and money away from that patient that could be used towards other, even more experimental options or going to clinical trials or other effective options. But at the end of the day, it's still the oncologist's decision, and they have to weigh, because we're not the experts. So they still have to weigh the patient's history, the other confounding non-cancer related factors that might affect how a patient's going to respond to any given drug or drug combination. And then for the patients, when they do see an effective or a high ranked treatment option, again, they have to talk to their oncologist. I will say that different oncologists, like any other industry, there's a spectrum of willingness to go off label or to try new things. And different oncologists will have different, risk levels of trying new treatment options. But ultimately they have to talk to their oncologist. We do often time and time again see that patients who advocate for themselves, who insist on, a deeper look at potential treatment options, they do tend to live longer and get more time than patients who just say, I'm just going to follow standard of care, whatever the doctor is going to give me. Like, that's just what I'm going to take. No questions asked. Whereas patients who do advocate for themselves, who do look at various different options, who are willing, unfortunately in this environment, willing to pay out of pocket for some of these things, they do tend to, not always, but, a lot of times we have seen that they they do get more time, than patients who just go with the standard of care.
Yeah, that's very true. And I did a podcast recently with the CEO of Manta Cares, and we talked about patient advocacy and advocating for yourself as a patient. And we know statistically that just even one email to your doctor that's advocating for yourself, just even one, speaking up just a little bit will provide that person with better outcomes. So I love what you're saying. I love ending with this message of advocacy and technology and thinking about what patients can do for themselves. This is a team. This is teamwork. Your oncologist is there to help you for sure, but you have to help your oncologist. And the more you're involved, and the more you find ideas to bring to your oncologist that they can evaluate, the better you might do. So I just want to thank you for all the work you're doing. It really gives me so much hope as a metastatic breast cancer patient myself. It gives me so much hope for the future. And I really appreciate everything you're doing and your time today.
Yeah, no, thank you for having me. And, thank you for, having this platform that sheds light on functional testing and, what we have to offer within that space.
And now an important announcement. All the information that was provided today was for educational purposes only. I'm a patient, just like you. I am not a licensed or accredited physician, therapist, or clinical researcher. All information provided is not intended as a substitute for the advice of a physician, therapist, nutritionist, or any other qualified healthcare professional.